All right. Hi, everyone. This is Clarence. I'm presenting a Eterna special group meeting. Yeah, let's get started. Today, I'd like to tell you all about a project that I've been working on to derive two-dimensional information on RNA structure from a type of chemical mapping experiment called mutational profiling. So you in the audience are likely already familiar with the concept of chemical mapping as a strategy for interrogating RNA structure. And these methods are especially useful because they can probe the base pairing status of each nucleotide in RNA, as well as telling us about where the structure may be dynamic. So in chemical mapping, briefly, the RNA of interest is exposed to modification reagents, which form adducts to the RNA based on the solvent accessibility or base pairing status of each nucleotide. And then the locations, the resulting modifications, are read out by reverse transcription, and the rate of RT stops at each nucleotide position is quantified and used to guide secondary structure prediction. So this sort of one-dimensional chemical mapping approach is, of course, the method by which we probe the constructs that you, the players, design in Eterna in each experimental cycle. However, there are limitations to what we call one-dimensional chemical mapping. For example, in a given reactivity profile, we can see that some residues are protected while others are exposed in the green and the red circles, but we can't necessarily pinpoint which residue a given protected nucleotide is base paired with. For example, this residue circled in green could be forming this magenta base pair, or it could be forming alternatively this green base pair, which would suggest an alternative secondary structure. So one way to resolve this ambiguity would be to make a point mutation at the protected residue and see which other residue or residues become more reactive since the base pair is now broken. So our lab developed this concept into an experimental protocol called mutate and map, in which we generate a library of single point mutants and map each of them. So here, as you make mutations along the y-axis, causing higher reactivity along the diagonal from the upper left to the lower right, there are correlated increases in reactivity. I don't think it's correlated to the position of the mouse. Is it? Oh, okay.
It's like that bug was fixed.
Awesome. Okay, yeah, so as you make mutations along the y-axis, you cause higher reactivity to appear along this diagonal, which is the location in the RNA that's being mutated. And then at certain mutation positions, you'll see correlated increase in reactivity along the x-axis. And so what's cool is that you can then directly see secondary structure elements, which appear as these cross diagonals because the base pairs are being broken along a helix. So this mutate and map technique allows us to get rich information about base pairing throughout an RNA. Now, the two-dimensional data from mutate and map experiments can be used to increase the fidelity of secondary structure prediction by algorithms like RNA structure. So for example, this tRNA, shown here, is both a critical type of RNA for biology and also relatively short. So we would hope that we would be able to predict its structure without too much trouble. And as it turns out, without any mapping reactivities, RNA structure fails to predict the correct structure. As you can see, there are these alternative base pairs forming in blue, whereas the native base pairs are shown in orange. Even with one-dimensional chemical mapping data, RNA structure still fails to predict the right structure, even though it's closer. But using mutate and map data gives you exactly the right structure. And beyond just predicting a single secondary structure, mutate and map can also give us a window into RNA structural ensembles, because individual mutations can sort of push RNAs into alternative secondary structures that may be less populated in the wild-type construct. So Pablo Cordero, who recently graduated from our lab, developed a program for using mutate and map data to parse the members of these structural ensembles. So here is an example from his paper pulling out multiple structures of the adenine riboswitch. So though mutate and map is a very powerful technique, it's not the easiest protocol to carry out because it requires generation of many individual sequences, which are probed in separate reactions and sequenced on the capillary electrophoresis machine. then those data need to be aligned and quantified. This means that it can't be carried out on native RNAs purified from cells, for example, and it can't be used on very long sequences because of the number of reactions that need to be done in parallel. We'd love to have a way to carry out the mutate map protocol in single reaction and to be able to probe native RNAs. So fortunately, over the last half-decade or so, revolutions in next-generation sequencing have allowed various labs, including ours, to start making chemical mapping even more highly parallel. So Eterna players will be familiar with this because that's actually what has enabled both the huge expansion in the number of RNAs that can be probed per cycle in Eterna, as well as enabling the use of the Greenleaf Labs RNA array to assess, for example, the thermodynamics of switch puzzle designs. So out of these recent studies, the last two, which are both from the Weeks Lab at UNC, are relevant to the work I'll talk to you about today because they suggest a fast way to probe native RNAs of arbitrary length and get correlated information. So the first study that I boxed on the previous slide presented an experimental advance toward avoiding biases that can occur during library preparation for sequencing readouts of chemical mapping experiments. So for example, the ligation efficiency of sequencing adapters to cDNAs for sequencing can be different depending on the nucleotide at the 3' end, which can cause problems for experiments that read out reverse transcription stop frequencies using paired end sequencing. But by adjusting the reverse transcription conditions, here using manganese in the reverse transcription and superscript 2, which is an older version of the reverse transcriptase that we normally use, the authors of this paper were able to cause the enzyme to induce a mutation in the cDNA rather than stopping at the location of the adduct. Then they can take the cDNA and sequence it, and then count the mutations to get an idea of how often each nucleotide is modified. So in their initial paper, the authors used ShapeMap to demonstrate similar secondary structure prediction accuracies between their method and capillary electrophoresis experiments. And then they performed some functional assays for newly predicted pseudoknots in the HIV genome. But essentially, this ShapeMap protocol is just a different way of doing the 1D chemical mapping experiments I described early on. But the most relevant part of the ShapeMap paper to my current work is actually here, where you can see that multiple modifications have occurred in a single RNA molecule. When multiple modifications occur in a single RNA, there's a possibility that the second mutation is correlated with the first mutation, giving higher order structural information, just like a mutated map. Now, the key advantage of mutational profiling is that then you can detect multiple mutations in a single RNA molecule, which you can't do with stop-based chemical mapping. And this circumvents the need to make an initial mutation prior to the actual chemical mapping reaction. So moving on to the second paper from the Weeks Lab, the authors used high doses of DMS, which is, if you recall, the other reagent that I showed you on one of my earlier slides, which modifies the base pairing edge, Watson-Crick edges of A's and C's, and they used a high dose of the modification reagent to potentially induce multiple mutations per RNA molecule, which are read out by the mutational profiling protocol. So the authors use their data in a couple of different ways. So first, they tried to infer what they call through-space interactions by calculating correlations between mutations that co-occur in their sequencing reads. And then they also did another analysis where they clustered groups of sequencing reads with similar patterns of mutations together to essentially pull out 1D mapping profiles for individual conformations. But what was most exciting to us about the study was the potential that this high-dose mutational profiling experiment could provide essentially mutated map data, which would solve several limitations of our current CE-based protocol, For example, like probing native RNAs or using a single mapping reaction rather than several 96-well plates worth, and also being able to probe RNAs of longer length. So we adapted, or basically performed their mutational profiling experiment on our own. And the overall workflow is what I'm showing here, which is first that you generate a DNA template, you transcribe the RNA, then perform the chemical modification, reverse transcribe it under mutational profiling conditions, and then sequence either sequence the full-length cDNAs, or perform a PCR amplification to amplify the full-length cDNAs, and then sequence that, which I'll explain in a later slide, and then perform paired-end sequencing, and then do analysis. So first of all, we were interested in seeing whether we could detect two-dimensional data from the data that the Weeks lab actually presented in their paper. So we found a data set in their supplemental data, which is essentially what's called a simple file that contains strings with a binary encoding with a 1 where there's a mutation and a 0 where there's not a mutation. And then Pablo actually wrote an initial analysis code to get to basically transform this simple file into two-dimensional data. So this is actually the data derived from their simple file, but presented in the manner of our mutant map experiments. So as you can see, this is for the P4P6 RNA. There are clear indications of secondary structure elements that actually correspond to the secondary structure elements within the P4P6 RNA. And then there are also punctate spots here that correspond to alternative tertiary structure base pairing interactions, such as the tetraloop receptor that is present in P4P6. And you might be familiar with that interaction because it's that tetraloop receptor interaction is what Joe uses for many of his initial RNA make designs. But now that we saw that we can actually find two-dimensional data within these high-dose mutational profiling experiments, we wanted to recapitulate the P4P6 dataset using by performing our own experiments. This ended up being a pretty long process because we were trying to track down the actual source of the correlated mutations. I'll show you a series of slides sort of covering the various experiments that were performed, and you should know that all of these experiments were performed by Anne, and it was a lot of work, so really grateful to her for doing them. One of the first comparisons that we did was to check whether we could see two-dimensional data just by sequencing the cDNAs compared to doing a PCR amplification of the full-length sequence. So here you can see that compared to the data set derived from the Homan et al. Data from their paper supplement, the data from PCR shows a lot less background than the data from cDNA, although the signal to noise is not very high and it's a little difficult to see the secondary structure elements. So this sort of makes sense because cDNAs are, if you sequence the cDNAs, they're going to include all of the various lengths of cDNAs because the RT will also stop within So while the RT does introduce mutations, it will also stop at modifications as well. And so when you PCR out the full-length sequence, then you actually avoid having these shorter products in your sequencing data. And then another hypothesis that we sought to test was something that Riju noticed, which is that there are actually chemical mapping correlated signals that involve G's and U's in the data. So this was actually kind of surprising because you'd expect that DMS can only modify at A and C residues. And so there shouldn't be mutations caused by adducts forming at G's and U's. And so another hypothesis that Anne tested in her sort of panel of experiments was to perform the modification reaction at a series of different pHs, ranging from 7 to 8 in different actual buffers, all the way up to 9 and 10, to see if the signal at higher pHs would more closely recapitulate the Homan data. And that's because the G's and U's can actually be modified by DMS if the pH is actually high enough. Unfortunately, it's sort of looking at these data, it's not very clear that any secondary structure signal is stronger at the higher pHs than at the lower ones. So another hypothesis that we wanted to test was to see if there was pre-modification of the RNA occurring before the actual chemical mapping experiment. And so this could potentially arise from various steps of the RNA preparation. So for example, during page purification of RNAs, it's common to use a UV lamp to shadow, in a technique called UV shadowing, to visualize the bands on a polyacrylamide page purification gel. You normally use a UV lamp to visualize those bands so that you can locate them and cut them out as part of the purification process. But as Riju and Anne showed in a paper a while back, actually, a high dose of UV actually generates modifications in the RNA at a pretty high rate, even for low exposures to UV light. So Anne tested whether the page purification would generate two-dimensional signal similar to the Homan dataset by using the UV lamp either held close to the gel, giving it a high dose, or held far away, giving it a low dose in the page purification process. We also tried using error-prone PCR to actually generate a mutation generate mutations in the DNA template before we even transcribe the RNA, which could potentially generate a mutation library that would then allow for more two-dimensional signal. And lastly, this condition here was actually to test whether or how similar the data from mutational profiling in the Weeks lab paper was to a standard mutate map experiment from our lab carried out using a capillary electrophoresis readout. So Anne actually performed the standard capillary electrophoresis experiment with the separate modification reactions and a separate reaction for each mutant in the library, and then combine them all
for the sequencing. She made the mutants all separately.
Oh, she combined them for the modification.
Yeah.
Right.
And then she pushed them to the same protocol.
Yeah.
So that library was guaranteed to have exactly sort of one mutant every 200 nucleotides.
Thanks. So you can see that the introducing mutations by various methods actually does produce some two-dimensional signal, but it's not sort of the same appearance as a signal from the Weeks lab mutational profiling data. For example, you can sort of, I guess it's a little bit hard to tell at this size of the plot, but in the UV exposures, you can see that there's some sort of blurriness in the secondary structure signal that's not present in the Homan et al. Data, which is very sort of precise, very similar to the mutated map, standard mutated map data set. Right. So carrying on from this experiment, I sort of was progressively developing an analysis software for our lab, which was separate from the Weeks lab software because parts of it were missing. So we couldn't carry out their exact analysis. And so as part of exploring these sort of various factors in analyzing the data, I found that reducing the mutation cutoff, so the number of mutations that's allowed in a sequencing read for it to be included in generating the 2D data set, I found that if I dropped it from 10 to 5, it actually appeared to clean up the our lab's data set quite a bit, but actually made the Homan et al. Data set look a lot more sparse than the data from the paper. So this sort of looks nice, but we would hope that the distribution of hits between our data when we analyze it and their data is the same. And actually, it's pretty similar. This is a plot of the frequency of the number of mutations per transcript. So within each sequencing read, you count the number of mutations and then calculate the frequency at which that number occurs. So taking their data set and comparing the mutation per transcript frequency to what they presented in their paper, you can see that it's very similar, sort of distribution between 0 and 10 in their chemical mapping condition, which is in red. And in our data set, we also saw a similar distribution with an additional set of sort of maybe broad peak of mutations per transcript that appeared at a little bit over 20. But this doesn't really explain why our data set looks distinct from theirs between why it looks distinct from theirs because so I might have expected that with that our distribution was different because our data looked much cleaner with a mutation cutoff of 5, but it actually looks the same. Is the upshot. So then I also tested whether deletions were actually potential potentially important source of the two-dimensional signal in their data set, in the mutational profiling data sets. And the reason is that the primers that we use to assemble the DNA templates for our RNAs are ordered from IDT, which is a company that synthesizes primers and oligos. But imperfections in the primers that we order could actually result in mutations or deletions occurring when we assemble the DNA template. And so I excluded deletions and found that it raises the background, these sort of strong horizontal striations a lot relative to including the deletions. Then given this result, we wanted to see whether we could reduce potentially the number of deletions that were occurring in assembling the DNA template by doing PCR from plasmids, which already include the full-length sequence of the template, and so they don't require bridging between separate oligonucleotides to make the full-length DNA. And so we performed another round of sort of searching for the signal in pre-modifications by performing PCR from the plasmid to generate the template versus PCR from PCR assembly, or doing PCR from plasmid, but with the introduction of mutations through error-prone PCR. And so you can see that the data for the assembly data set looks pretty similar, and the TAC condition also gives signal similar to the signal that we saw in the TAC condition for the previous set of experiments. And PCRing from the plasmid appears to give a slightly weaker signal to noise for the secondary structural elements, which you can sort of vaguely see if you squint hard and sort of know where to look.
But also some of the hits are in different places in the plasmid versus the assembly. We can probably point to a few, like like right there.
So here,
those hits are only present in the plasmid. Around the same region, there are hits in the assembly, but they're in different spots,
right, suggesting
that those are arising. At least those are arising from
the difference in
the DNA, because that's the only thing different.
Right. Yeah, that's true. And then sort of to further explore the effect of
one thing that's interesting is that there's some other hits that are in the same place. Mm-hmm. And those hits, I think, in the original analysis from the Weeks lab, they focused on signal at A's and C's because that's where DMS usually hits. Right. And I think that's totally kosher. It's great. Mm-hmm. And I think that that signal may actually be due to double DMS hits in the RNA as opposed to an imperfections in the DNA. Yeah. Right. So it's kind of worth pointing out that there may be two sources of interesting signal in these data sets. Yeah. One of which was discussed before, and it may well be due to DMS hits. Right. But the other one is something that is only being uncovered now and could be due to what you think would be bad, which are imperfections in the DNA library. Right. Yeah. Cool.
Yeah, so I also did an analysis where I measured the overall modification rates over the P4, P6 sequence. And this was basically just to confirm the sort of initial and sort of the amount of modification that was occurring in the template itself versus a condition where the RNAs were not treated with DMS versus the condition where RNAs were treated with DMS. So that's template, nomad, and DMS here. So you can see that so in the nomad data, the error-prone PCR shows a much higher modification rate over P4, P6 than either assembling or getting the template from the plasmid using fusion, which is does not introduce which is not error-prone, or assembling the DNA template using primers. And then when you perform the DMS modification, they all shoot up to very high modification rates, which is what you expect. There was one strange thing from the result here, which was that in the template for the assembly, the PCR assembly template, we actually saw a lot of modifications and surprisingly actual like two-dimensional signal, which I really have no idea where that could come from because there's no reason for there to be correlated mutation, like structurally correlated mutations in a double-stranded DNA template.
Maybe we should check if those are at the primer boundaries.
At the primer boundaries? Oh.
The assembly, yeah. Let's talk about that.
I don't have the plot, but...
Have you made a similar plot of modification rates at G and U positions versus A and C positions?
No, I haven't.
That might be worth doing. I think that one prediction, if the model like says we were working on, if our working model is correct, the G and U position should not show enhanced signal in the DMS versus nomad conditions. It's easy. Just remake this plot over just A's, just C's, just G's, just U's. Yeah. Sum up with them.
Okay. Do that.
Cool.
Yeah. But this also potentially supports what Riju said, which is that there's like a baseline level of DMS-based modification, but then another level of like correlated mutations that occur from the way that you get the DNA template, which could be similar between doing it for getting the template from the plasmid or from PCR assembly. So more recently, previously the map2D analysis software was aligning read 1 and read 2 separately to the wild type reference sequence and then counting mutations that way. So one difference between our analysis at that stage and the shape mapper pipeline is actually that the shape mapper performs sequence alignment and then combines the reads for comparison to the wild type. And so something that I did more recently was to use the software called BB-Merge, which is part of this BB-Map software suite of bioinformatics tools, to actually merge the read 1 and read 2 FASTQs together before performing the alignment to the wild type sequence. And so the results are shown here. On the right, upper right, is the data for the initial data for one specific experiment, which was using P4, P6 from the PCR assembled DNA template, and then I took the same data set and pre-merged the FASTQ files and performed the analysis, and then also ran the shape mapper pipeline, but it's not the entire shape mapper pipeline because we're missing one script that converts a certain style of mutation recording into the sort of simple format that I described to you earlier. And so I had to write that part myself, and it basically takes, does a direct conversion of this particular style of mutation recording to the simple file, but there are a couple of parameters that it could vary. And so this data set here is shown with sort of the best conditions or settings for my script to convert their mutation strings to simple file. And in comparison with the new pre-merged map2D dataset, it looks actually pretty similar. There's even sort of background, potentially noise in some of the same regions between the two. But both of these are pretty, they're better than the original analysis without pre-merging the reads, that you can actually see in comparison to the Homan et al. Supplemental data set that they're pretty similar. So again, I did a quick analysis of the frequency of mutations for transcript to sort of compare how similar the data sets were after analysis. And you can see that from running sort of shape mapper with my hacky conversion to simple file, the map2D with pre-merging of the two FASTQ, the two reads, they look pretty similar, but there's also a large amount of reads in the map2D analysis that are currently basically thrown out or have the mutations assigned to be 0 because, for example, there are too many deletions within the read, which suggests that the RT actually skipped from one place to another inside the RNA when it was going, and so that information is not that useful. But you can see that the distribution of the map2D analysis with the pre-merged FASTQs is more similar to the shape mapper-based analyses than.
Have you looked at those jump reads, the ones that jump? Have you plotted those in 2 dimensions to see what they correspond to? Beginning and end of secondary structure elements?
No, I haven't done that.
That's really cool. In fact, yeah, both without DMS and with DMS. Yeah. Maybe the RT jumps no matter what, but something maybe the DMS forces the RT to jump and then it. Let's see. Forget what I just said. Yeah. But you can look at, we also have the UV condition. So maybe the UV forces the RT.
Oh yeah, I can see if there's more gaps.
So I think there might be a really cool signal there.
Okay, yeah. And finally, I also made another discovery recently, which is that the Weeks Lab deposited their sequencing data actually on sequencing read database, the SRA database, which you can actually download the FASTQs from there. And so here I'm showing, I downloaded the FASTQs for their P4, P6 with magnesium conditions, which are essentially the same as our P4, P6 conditions that I've been showing you this whole time in the bottom row here, and also their FASTQ data for RNase P plus magnesium, which is one of the RNAs that they probed in their paper. And so in the left column here, I'm showing the map2D analysis just starting from the simple files that the authors provided in the supplemental data of their paper. And then in the middle, I performed the map2D analysis with pre-merging read 1 and read 2 for RNase P. For P4, P6, they actually, for some reason, it was only unidirectional. They didn't have paired end data for it. I'm not sure why. And then I also ran shape mapper on both of these data sets. Yeah, you can see that the map2D analysis of the RNA-seq fast queues actually looks pretty similar to the simple file from their supplemental data. And there may be something, I'm not sure why so few of the reads from the fast queues were actually properly counted as part of the 2D data set when I used the shape mapper analysis. But I can track that down.
I think what's more important to track down is the bottom row.
Yeah, right. So well, so I'm not sure if, yeah, I guess it's the discrepancy between the data from the map2D versus shape mapper and the simple file suggests that my way of analyzing with using just one fast queue might be faulty.
There might be a bug. It should be easy to fix that because there are these big artifactual hits coming out of this pipeline, the map2D pipeline, that aren't present together. So you can just go look at those specific lines in the fast queue file and see where they didn't pass the filters, where they passed your filters but not the other, the shape mapper filters. So that's,
because both of these have fewer reads included.
And now you can tell. Now you have like a guide, a signal to follow. Yeah, so that's very exciting. Yeah. Then you should probably go back and reprocess all the previous data sets, like the pH and all that.
Yeah, that's what I intend to do.
But exciting, yeah. That's great.
All right. I also did this analysis of the frequency of mutations per transcript. You can see that in the RNA-seq, there's overall greater number of mutations per transcript, which you might expect since it's a longer RNA, so there's more sites for it to be modified. Right. I'm not actually, I forgot to show the panel from the paper showing the correspondence between the distribution of mutations per transcript, but I think it matches up again.
So you can see that the map2D pipeline is producing a bunch of other garbage. It should be really easy to figure out what's going on.
Yeah, all that stuff.
Yeah, but that's exciting. That's so close.
Okay, so yeah, overall, sort of the main thing that allowed us to get to a better data set was pre-merging read 1 and read 2. It's still not entirely clear what's causing the 2D signal, but as we discussed, there are ways that we can pull that out by comparing the GNU modifications to the ANC modifications, for example. I also will use the RNA-seq data from the map2D analysis compared to shape mapper versus a simple file to guide RNA structure predictions. So previously, SITC did analysis and showed that using 2D data derived from the simple files by the Weeks lab for RNA-seq actually greatly improved the secondary structure prediction. So hopefully the map2D data will be able to do similar. Then I've also started doing the mutational profiling experiment myself based on Anne's extensive notes on how to do it on the L21 group 1 intron, which Anne actually included in in one of her previous rounds of experiments, but we were sort of stymied by a mispriming event in the PCR amplification step. So I guess I want to leave you with this, which is just that I have already reverse transcribed the RNAs. The results look pretty similar to the results that Anne saw in her previous experiment. But hopefully the PCR amplification will look fine, and then I can sequence it and actually get 2D data.
Oh, cross our fingers.
Oh, yeah.
If it doesn't look fine, you need to purify out the full
Yeah.
of size selection. Yeah, that's great. Okay. Are there any questions from Eterna?
Great. Thanks.